Alright, Bismillahirrahmanirrahim and Assalamu alaikum students. Welcome to our second online session for the course Biot-710 Advances in Bioinformatics for the semester Spring 25. Today is June 21, 2025. In last session we discussed about the section which involved course contents for the databases, data format, database searching, and all related to that. The second section of our course comprises of bioinformatic analysis involving protein sequences. The essentiality of this particular domain of bioinformatics is that overall, although major research work in bioinformatics is being done in the domain of nucleic acid sequencing and analysis, but as we are progressing more towards the annotation of protein sequences, we are getting more inside knowledge of the function and structure of the proteins, which are actual the working force of the cell. Nucleic acid is not basically the actual working machinery of the cell; it is just a blueprint. But what the protein which are being transcribed from those nucleic acids, they are actually trying to transport the nutrients. They are replicating the DNA. They are dividing the cell. They are performing some very essential tasks like providing a cytoskeleton to the cell. They are present in the membrane as an integral and peripheral proteins. They are transporters which are involved in uh, bringing in uh, and exporting out the required molecules. So all these activities are governed by the protein sequences. So uh, as as closer we get to the function or structure of these proteins, we better get a deeper insight knowledge of the cell functions. So that's why, although there are also tools which involve analysis of nucleic acid sequences, but s- those involve much computer programming aspect, and secondly, they are more prone to error as compared to protein sequences. That is actually my assumption. So what we need to understand that when we are trying to get a beginning perspective of this domain of bioinformatics, so we will get a better understanding while studying protein sequences as compared to the DNA sequences. And the first aspect which we can understand about the uh, protein is that its structure. Definitely, protein sequence, we have 20 amino acids, and if you recall that protein structure has four levels: primary, secondary, tertiary, and quaternary. So, primary, the sequence of protein is known as its primary structure, which incorporates 20 essential amino acids, not essential, but 20 standard amino acids are lined up uh, using the peptide bond in a different combination, which makes up the sequence of the protein, and that is actually translated by the gene sequences which are present in the form of open reading frame. What is open reading frame? uh, which has a start and a stop codon within the DNA sequence, इसको हम open reading frame कहते हैं. Secondary structure is the hydrogen bonded three dimensional local conformation. So यहाँ पे हमारे पास कौन सी bonding involved है? Previously we had the peptide bond. In primary structure, secondary structure में we have the hydrogen bond. The two most common secondary structures, that means के इसके अलावा भी मौजूद होते हैं, but two most common कौन से? Alpha helix and beta plated sheet. In addition, from other commonly occurring secondary structures, इसके अलावा ये तमाम secondary structures भी मौजूद हैं. रैंडम कॉइल्स भी होते हैं. सो ये तमाम के दरमियान में हाइड्रोजन बॉन्डिंग होती है दैट मेक्स देम अ सर्टेन स्पेसिफिक शेप. देन वी हैव द कैरेक्टरिस्टिक्स ऑफ अल्फा हेलिक्स. इसका रेडियाई जो है वो 2.3 एंगस्ट्रॉम है विच इज़ मल्टीप्लाई बाय 10 एक्सपोनेंट माइनस 10. अगर इसी फिगर को मैं नैनोमीटर में लिखना चाहूँ तो दैट विल बी 0.23 नैनोमीटर. In a right-handed helix means के it is rotating anti-clockwise with 3.6 amino acids per turn. ठीक है, जो कि 360 डिग्रीज होते हैं इन अ कंप्लीट सर्कल. तो जब 3.6 टर्न्स होंगे तो डेट विल मीन 100 डिग्री टर्न्स पर रेजिड्यू. हाइड्रोजन बॉन्ड, कार्बोनाइल बॉन्ड, अमाइनो ग्रुप्स, ये तमाम इनकी करेक्टरिस्टिक्स आ जाएंगी. फिर अल्फ़ा हेलिक्स की जो अल्टरनेट फॉर्म्स हैं, 310 हेलिक्स, इनकी करेक्टरिस्टिक्स हैं, इनको रिव्यू कर लीजिएगा. आई वुड से कि इनको आप लिस्ट डाउन कर दीजिए कि कौन सा लाइक टेबलेट फॉर्म कि कौन सा सेकेंडरी स्ट्रक्चर है, उसकी क्या करेक्टरिस्टिक्स है, कितने रेजिड्यूज हैं, कितना रेडियाई है, एटसेट्रा, और इनकी प्रिवलेंस इन प्रोटीन्स कितनी है. देन वी हैव कॉइल्ड कॉइल्स which is also known as super secondary structure. यानी कि दो secondary structures आपस में in combination forms a super secondary structure. Each strand of a coiled coil has a repeat of 7 residues A B C D E F, then again A B C D E F. इस तरीके के 7 डिफरेंट अमीनो एसिड के कॉम्बिनेशन का रिपीट्स मौजूद होते हैं. Good candidate amino acid of these positions are isoleucine, leucine, and valine. तीनों hydrophobic kind of amino acids, non-charged. In contrast to the helices, a beta-plated sheet or beta sheet involves two or more polypeptide chains, and the hydrogen bonds are formed between residues that are part of different polypeptide chains. ठीक है, so ये within a polypeptide chain hydrogen bonding 1 to 4th, second to 5th, थर्ड टू सिक्स अमीनो एसिड इस तरीके से होती है. और जो बीटा शीट है वो डिफरेंट पॉलीपेप्टाइड चेन्स के दरमियान में हाइड्रोजन बॉन्डिंग से एक शीट, एक बिल्कुल प्लेनर फॉर्मेट का स्ट्रक्चर सा बन जाता है. Do we have any questions? No. And we have 14 students up till now. Okay. Then we, after these some types of secondary structures, we have the tertiary structure of the protein, which is formed by interactions between the side chain are groups such as the ionic interactions, hydrophobic interactions, hydrogen bonds, and disulfide bonds. So these four kinds of different interactions are formed between the side chains, and these are forming the tertiary structure of the proteins. इस फोल्डिंग प्रोसेस के लिए जो सेलुलर मशीनरी इन्वॉल्व होती है दैट इज नोन एज शिप्रॉन्स molecular shiprons. In folded conformations, most proteins contain specific domains that are discrete structural and functional units of the proteins. And at the end, we have the quaternary structure, which actually um, is um is made when the protein has at least two subunits. When the protein has a monomer in nature, then the last structural level is only the tertiary level. But if two monomers of the same proteins join, or different proteins like homodimer, heterodimer, or homotrimer, heterotrimer, homotetramer, heterotetramer, in all these aspects they will be comprising of a quaternary structure of the protein. Then we have the properties involved in the protein bonding, which is amide linkage, as in the figure we can see that we have an amino acid like this, okay, and this carbonyl carbon and this amino group is actually the peptide bond. And similarly here, this amino group and this carbonyl is the peptide bond of the another amino acid. This peptide bond is planar, means it does not rotates on its axis. That's why they have shown that in a yellow background. But the ones which can be rotated are these bonds, amino group and alpha carbon, and this bond is known as phi bond. In carbon alpha and carbonyl, this is known as psi bond. These two bonds are involved in the rotation, and this is a Ramachandran plot which shows the accepted regions or allowed regions where the r- degrees of the angles of these psi and phi bonds can exist. And we can see that not all, from 1 minus 180 to plus 180 of the phi angles, and same for the psi angles, not all region is shaded. यहाँ पे certain regions हैं जो कि हमें allowed regions बता रहे हैं for the uh, beta helices and alpha helices. और structure 3 में one of the most important property of the elements which atoms which are being under observation in the protein structure, which is the van der Waal radii, which is the radii uh, under which it has some influence on any other atoms that can be repulsive or attractive, but it may have some force on any other atoms. रामाचंद्रन प्लॉट इसकी डिटेल्स हैं कि किस तरीके से हम साइन फाइंगल्स की करेक्ट वैल्यूज़ इससे आइडेंटिफाई कर सकते हैं। एंड एग्जांपल्स के तौर पे इन्होंने एक प्रोटीन्स के साथ इसको बताया गया है। दिस रामाचंद्रन प्लॉट इज़ अवेलेबल एट उपसला सर्वर। इसके अलावा प्रोटीन स्ट्रक्चर के अलावा भी कई सारी प्रॉपर्टीज़ हैं दैट कैन बी डिटरमाइंड यूजिंग बायोइन्फॉर्मेटिक टूल्स, ठीक है। So we have physicochemical properties of the protein using XPASI server of prot param. So what ProdParam does, in this screen we can see the results. It determines if we put submit the sequence of the protein. Not necessarily it's a known protein, it can be any unknown protein. It will determine its molecular weight, its theoretical isoelectric pH, atomic amino acid composition, number of amino acids, uh, extinction coefficient, estimated half-life, instability index, aliphatic index, and a grand average of hydrophobicity, which is also known as GRAVI. What is the extinction coefficient? If you can see here, indicates how much light is a protein absorbs at a certain wavelength. ठीक है, so ये एक बायोफिजिकल प्रॉपर्टी है. और every protein will always be having a specific extinction coefficient, and any other protein, despite having same amino acid numbers, not the composition but numbers, but since the composition will be different, so it will always be giving a different extinction coefficient. So this particular uh, property, we can identify an unknown protein among a pool of many proteins. What else properties we have? Aliphatic index is like a protein having aliphatic amino acids. Those are hydrophobic amino acids. Instability index present an estimate of the stability of the protein in a test tube. Gravy is something related to again hydrophobicity. So there are regions in the cell which are hydrophobic, for example, the membrane. So a protein having a high gravity value will most definitely be present in the membrane region. For every these scientific words, you must be aware of its definition, and if there is a bioinformatic tool which can identify these properties of a protein. Prediction of post-translation modification and protein sorting. Again, these are two properties. These properties of a protein which can be identified using the bioinformatic tools, and also this will help us to get a better insight of the protein function. These are the names of the tools by which we can identify these two characteristics. Secondary structure prediction. So there are methods which can identify the secondary structure of the protein. Initially, there was a method known as Shaw, Fishman, Fessman, and GOR methods, which basically calculates the probability of certain amino acid among the protein which has been involved in forming any specific secondary structure. So using the probability values, and then after that, if there is a certain patch of amino acid present in a certain region of the protein, we can estimate or we can hypothesize that this particular region of a protein may be involved in forming some secondary structure. But the advanced forms of the secondary structure tools involve homology modeling and protein threading. Homology modeling works on the principle of protein alignment and sequence similarity, while protein threading more specifically worked on fold recognition. So every amino acid having a certain aspect effect on the neighboring amino acid, and this actually keeps on calculating, and at the end we end up in forming a protein structure. But that protein threading involves a very high computation, very long calculation, so it requires supercomputers to perform those tools. These are the name of the tools by which we can identify the protein secondary structure. The next, okay, this one is the prediction of the accuracy Q3 score, which identifies the quality of the predicted structure of specifically helix, strand, and coils. That is done. That is why it is termed as Q3. और इसकी values होती है from point 0 to 1, and one being the perfect or 100% prediction. Prediction of domains and motifs. A domain is a part of the tertiary structure of the protein. Each domain is a discrete globular unit that folds uh, independently of the rest of the protein. So overall protein में there are structural regions. So उनकी existence जैसे जैसे पता चलती जाएगी, so we can get better understanding of the function of the protein. Its का size होता है 20 से 25 amino acid, and then again there are tools which can identify these domains and motifs. So there are some examples of domains like SH3, which is C homology 3 domain, which is around 50 amino acids and are involved in protein-protein interactions. The chromatin organization modifier domain, which is 30 to 70 amino acids and involved in the assembly of the protein complexes or chromatin. So, not necessarily we get information of the all protein, but in that protein of, let's say, 400 500 amino acid, उसमें से हमें इन 30 40 से सीक्वेंस अमीनो एसिड का भी अगर फंक्शन या डोमेन पता चल जाता है, तो then we can identify the or relate its function with these properties of the domain. Then we have the motive. Motive क्या चीज़ है कि does not fall independently of the rest of the protein, which has a sequence motive or structural motive. A smaller patch of amino acid amongst the domain, which is actually the functional part of that domain. We have the tool known as interproscan, which identifies the domains and motifs within the protein. We have the tool known as CDD, Conserved Domain Database. It has information of some domains, and we can do a particular CD search to identify domains and motifs of some hypothetical proteins. Transmembrane helix prediction. again we have a tool which can predict a protein is being present in a membrane or not. The tool name is known as I think rhythm. Viewing the 3D structure of the proteins, we have the database of PDB and protein database may we have submitted structures of the protein using the methods of X-ray crystallography and NMR spectroscopy, and now the number of electron microscopy structures are increasing. And as of July 30, 2013, number of structures are 92,000. Now they have increased, I think, more than 200,000. We have tools known as First Glance in JMOL, which help us to identify the structure of the protein. Allergenic protein databases, substances that causes allergic proteins are called allergens. ठीक है, इम्यून सिस्टम में जो एलर्जिक रिस्पॉन्स होता है प्रोडक्शन ऑफ आईजीई इम्यूनोग्लोबुलिन ई दैट इज आफ्टर an allergen is being entered into the body. So वो स्पेसिफिक प्रोटीन्स कौन सी इन्वॉल्व हैं उसके लिए दी वी हैव द डेटाबेस nomenclature of writing the allergen, the protein. Structural database of allergenic proteins SDAP. Linear epitopes, conformational epitopes, and allergenicity. So, what epitope is a part of the antigen which involved in the induction of the immune response. So, the cognate antibody binds to the allergenic epitopes to trigger the allergic response. Epitopes can be a linear or conformation. In a linear epitopes, the amino acid sequence is continuous, whereas in a conformational epitope, the 3D conformation of the protein brings two separate sequences at least together to create the epitopes. ठीक है, या तो वरना सिंगल सीक्वेंस आपके पास एपिटोप एग्जिस्ट कर रहा होगा या फिर दो डिफरेंट सीक्वेंसेस से आपके पास वो पर्टिकुलर सीक्वेंस आ रही होगी to generate an epitope. An exact match of six contiguous amino acids and overall sequence identity of more than 35% in a sliding window of 80 amino acids. Any protein that satisfies one or both of these criteria should trigger uh, additional investigation to confirm whether the protein may have allergic potential. So these are some criteria which actually we need to understand कि बायोइन्फॉर्मेटिक टूल्स काम कैसे करते हैं. ठीक है. यहाँ पे लिखा हुआ है कि जो क्राइटेरिया जो पैराडाइम है उस पे कि एग्जैक्ट मैच ऑफ सिक्स कंटीन्यूअस अमीनो एसिड्स. ठीक है. या तो छः अमीनो एसिड ऐसे जो लगातार एक एलर्जिक डेटाबेस पे जो सीक्वेंस मौजूद है वही हमारी सबमिटेड सीक्वेंस में मौजूद हो, या फिर जो सबमिटेड सीक्वेंस है उसकी सीक्वेंस सिमिलैरिटी हमें मिलती है 35% विद दी अवेलेबल सीक्वेंसेस इन द डेटाबेस. अब ये चीज़ कि ये एक ऐसा क्राइटेरिया है कि जिस पे अगर वो पाँच हो जाए तो नॉट ऐसा नहीं हो सकता कि वो बिल्कुल एलर्जिक नहीं होगा. पॉसिबिलिटी हाउ. या फिर 35 से 34 हो जाए तो अगेन देयर इज़ अ पॉसिबिलिटी कि वो बिल्कुल भी नॉन एलर्जिक होगा, नहीं हो सकता है. लेकिन these are the standard values which are being observed during the uh, uh, during the investigation in the database कि ये चीज़ें अगर हम रखेंगे तो definitely we will be ending up in an allergic protein. We have allergenicity prediction servers. अब ये बुक अगर हमारे पास 2015 की है which is almost 8 years ago, तो not necessarily we will be having these servers till now. But again एक चीज़ मैं बताता हूँ कि बायोइन्फॉर्मेटिक टूल्स, almost all tools which can be numbered in thousands and even millions, they work in the same function. We submit a sequence, we set up parameters, we decide an algorithm, we perform its computation, processing is done, some initial results are generated, and using again computation, we analyze those results. Intrinsically disordered protein analysis. What it is are characterized by the lack of stable tertiary structure under physiological conditions. Okay, but the name indicates it means it seems that these proteins are disordered, so they may not be functional, but actually they are physiologically functional proteins, and they have some disordered region, which we will be studying what it is. The lack of structural order in a protein goes against the traditional wisdom that protein function depends on its stable tertiary structure. ठीक है, which is a structure function paradigm कि प्रोटीन का स्ट्रक्चर स्टेबल होगा तो ही वो काम करेगा, अदरवाइज नहीं. बट देर में आर सम प्रोटीन्स जिनमें ऐसा हमें नहीं देखने को मिल रहा. It has long been realized that proteins possess configurational adaptability, that is the induced fit model. Proteins इस तरीके से भी functional role play करती है अपने conformation को change करके. However, the presence of disordered segments in a functional protein became apparent when the crystal structure of various proteins becomes available. Techniques such as NMR, X-ray, and the circular dichroism helped uncover the disordered and unstructured state of certain proteins. ठीक है. इन टेक्निक से हमें पता चला कि ओवरऑल प्रोटीन के 3D स्ट्रक्चर में देर आर सम डिसऑर्डर्ड रीजन्स जिनकी वजह से वो प्रोटीन हमें एक फंक्शनल रोल परफॉर्म करती है. और वो कैसे अनस्टेबल या डिसऑर्डर रीजन पता चला कि उनके जो इलेक्ट्रॉन डेंसिटी थी वो एक जगह पे नहीं रुक रही थी, वो काफ़ी डिस्टर्ब थी. एटलीस्ट 50% ऑफ द यूकैरियोटिक प्रोटीन्स, यहाँ पे देखें प्रोकैरियोटिक में यूकैरियोटिक प्रोटीन्स का जिक्र है, पोजेस एटलीस्ट वन लॉन्ग that is greater than 40 amino acid loop, while this fraction is low in prokaryotes and archaea. Protein disorder is found within the loops. Coil may also assume disorder as they only assume globular structure when the coil proteins interpret with one another. DNA RNA बाइंडिंग एंड जीन रेगुलेशन, ये डिफरेंट प्रॉपर्टीज है जहाँ पे ये पर्टिकुलर डिसऑर्डरनेस फंक्शन रोल प्ले करता है. IDP plays an important role in signaling and regulation. They are much more abundant in eukaryotes and prokaryotes. ठीक है. सो ये बड़ी इंटरेस्टिंग इन्फॉर्मेशन है. ओवरऑल स्ट्रक्चर में अगर मैं इंस्टेबिलिटी भी दिख रही है तो हमें देखना पड़ेगा कि ये इंस्टेबिलिटी प्रोटीन का गलत प्रेडिक्टेड स्ट्रक्चर है या फिर ये कोई आईडीपी है. So for that we have IDP databases, which one is the disprot. Again, we submit the sequence, we get prediction, and then we analyze the results. ठीक है, that is all for today's online session, and इनके डिटेल्स को मज़ीद हम डिस्कस हम अपनी मंडे वाली क्लास में कर लेंगे. Let's see if we have any questions. No questions in chat group.